Our next presenter is Dylan, that is a coding superstar. I don't know how else to define him after reading his resume when he sent it for applying for the internship. So he's the VP of a programming and coding club, and he also recently won an award at the International Science and Engineering Fair for his project in computational biology. It is so complicated, I need like the thing to read. I don't know how you manage to win an award there. So next one, Dylan.
Hello, everyone. My name is Dylan, and I'm a rising senior at Dreyfus School of the Arts. And during these 6 weeks, I've had the privilege of working at the Bidea Lab as a scientific programming intern. And my project is modeling the male adult nerve cord of the Drosophila melanogaster using spiking neural networks. And I know that sounds like a mouthful, but essentially my project is translating the neural connections within the ventral nerve cord of the Drosophila melanogaster, which is the fruit fly, into a computational model using what we call spiking neural networks. So why the Drosophila? To understand why we use the Drosophila, we have to understand what is connectomics and why we even study it. So connectomics is the comprehensive study of all synaptic connections in a nervous system. And since we have to map all the neurons and neural connections, an animal with a really simple nervous system, such as the Drosophila, is perfect while still allowing us to study neuronal locomotion. Its stripped down nervous system means there's only 23,000 neurons in the ventral nerve cord, or VNC, which is the body part of on the fly equivalent to the human spinal cord. As one of the previous interns, Jacob, mentioned, humans have exponentially higher amounts of neurons with 86 billion neurons in the brain alone. So there would be an exponential amount more of neural connections. As for why we use the ventral nerve cord, it's because of the recent data on the male adult nerve cord, or MONC, that came out around 2 weeks before my internship. We've been able to map the Drosophila's brain before, but using the ventral nerve cord is an opportunity that we've only recently gotten due to this new data set. So to first model the VNC, we have to first understand how neurons work in the nervous system. And this requires a little bit of electrical engineering, but I promise I'll keep it simple. So essentially, we can think of the nervous system in any animal as a computer. And there's signals constantly firing back and forth in that computer. So if we can think of the nervous system as a computer, we can think of the individual neurons in the nervous system as a circuit. And on the left side here, I've modeled a little graphic of a circuit for you. We can see electricity flowing in and out of the circuit with an R standing for resistor and C standing for capacitor. The resistor essentially regulates how much electricity can come in and out of the circuit, and the capacitor stores charge to be used at a later time, like a battery. So essentially, I show you this circuit because circuits work, neurons model just like circuits, and using our computational model, we can model our neuron just like the circuit. So now we have to find an appropriate model that uses this electrical circuit behavior. For this, we use the leaky integrate and fire model, or LIF. LIF models the individual neurons firing and its action potential. Leaky stands for the fact that a cell membrane is semi-permeable and the neurons slowly leak out charge even though they're constantly intaking charge. Integrate stands for the accumulation of signals from many presynaptic neurons, essentially meaning that since there's many presynaptic neurons coming into one neuron, the electrical charge builds over time and accumulates, it integrates. And then we have fire, which is the action potential. This is the released energy from a presynaptic neuron that fires to the next. As we can see in this graph here, we can see all three of those principles demonstrated. First, we can see that this is a graph of an individual neuron and its firing and its cell membrane potential with on the x-axis is time and the y-axis is the voltage. And we can see at 0 here, this is the resting state. This is the neuron doing nothing, it's just sitting there. And then we can first see our first electrical signal coming in, and that's that spike here. And we can start to see it decays towards 0. This is because even though it's received an electrical charge from a presynaptic neuron, the charge slowly leaks out of the cell membrane. So it decays, it leaks. And then we can see the idea of integrate here. We see multiple kind of dotted lines here, meaning there are multiple presynaptic connections coming in and all the charges are adding up together. And then eventually when they add up together and reach the threshold, there's a huge spike in voltage known as the firing of the action potential. And that's telling the neuron, hey, we have enough signals, we can fire to the next neuron. And that's a huge voltage spike here, followed by a reset back to its resting potential. So now that we understand how each neuron fires and communicates, in what form does our data come in? Well, we can see that our data comes in a form of a table, and I've mapped out the table right here. So essentially, we have presynaptic neurons, the neuron firing, postsynaptic neurons, the neuron receiving the signal, as well as synaptic weight, which is how strong the connection between those two neurons is, and modified synaptic weight, whether that connection is inhibitory or excitatory. And that's determined by our predicted neurotransmitter. So essentially here we can see that there's a presynaptic neuron with the ID 10,409 firing to postsynaptic neuron 25,042 with a synaptic weight of 2 and a modified synaptic weight of -2. Well, that's kind of a lot of words. The idea is that this one row represents one unique connection between two neurons, and that extends 5.3 million more rows in what we call an adjacency table. And for researchers to parse through this table normally would be extremely difficult. You wouldn't be able to tell what neurons connect to what, and then what neurons do those connect to. So we need to build a model to actually interpret all of this data. So how does this come together? Now that we have our data and our lift model per neuron, we need to put together the network. Each neuron will have its own differential equation to model the lift function, with each lift function being put together into what's known as a spiking neural network. We model it with the membrane-Volter equation of a dV/dt. And essentially, this spiking neural network of neurons firing back and forth at each other and communicating is what our mapped connectome looks like in our computational model. So now that we kind of have the architecture of a model and we've established what data is coming in, what does the workflow look like for a researcher who's going to be using it? So essentially, first we see here that we're feeding in the Monk V1 data, which is essentially the male adult nerve core data, as well as researchers' custom neuron names. This is researchers going into the Monk data set and picking out neurons they'd like to focus on, ones they'd like to silence or stimulate, as well as ones they'd like to rename into more user-friendly names instead of the 5-digit IDs that we previously saw. And then afterwards, we do a remapping of the IDs into our own internal index from those 5 digits into our own internal index that helps process the model better. And then we run the simulation and record spike data. It's to be noted that these two processes are done and integrated via the programming language Python. And then we process our results into 4 main graphs. So what happens if we run the model in a real-world scenario? So in the Bidet lab, we focused on 2 neurons, 10,564 and 10,788. These are neurons that are known as the front leg stop neurons, and they're depicted here. These neurons are responsible for halting the Drosophila's front legs, and we stimulated them to see how the local circuit reacts. We can see raster plots generated from the data that are very dense, and these raster plots obviously show us that we're stimulating these neurons at a high rate, but that doesn't really tell us much because these were the neurons we picked out and stimulated artificially. But then we can go to the heat maps and we can see the stimulation rates of the top 20 neurons in that local circuit. And we can see that front left and front right are the most stimulated. And again, that doesn't really tell us much, but we can also see that there are other neurons such as SAP1 and SAP-SUN1, which are also highly stimulated. And then we can investigate further into those specific neurons with this tool. So we've compiled our data into graphs for data scientists and kind of researchers, but here's a more intuitive way of kind of picturing it, so using nodes. So the first thing we can do is plot the nodes of all of our neurons. So essentially, these represent all of our neurons, and they're proportional to size. So the larger the neuron, the more it's being stimulated in our kind of experiment. And then we can plot our excitatory connections. So these are connections that are simulation generated of excitatory connections between neurons, and they're proportional to the width of the neuron. So essentially, proportional to the width of the arrow. So essentially, a wider arrow means a stronger connection. And then we can plot our inhibitory connections. These are also proportional in strength. So what does this data kind of tell us? Well, we know we stimulated the front neurons here and here. We see other neurons with high stimulation rates evidenced by their large node sizes here, 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 and here. So these neurons are identified as SUN1 right and left and SAP1 right and left. We can attribute these high stimulation rates with the massive excitatory connections in green right here and here, as well as here and here. So what does this tell us essentially? So when we're looking at the neurons and we're looking at the predicted function of the front left and right neurons, we understand that once stimulated, they are meant to halt the front legs of the Drosophila. So when they're stimulated and we see other neurons highly stimulated in the local circuit, we understand that these are neurons that the front left and front right neurons are essentially recruiting in order to help it stop that motion. And we can verify that with our adjacency table seen previously and see that the front left neuron right here, it connects to the SUN1 right neuron right here with a strong synaptic strength of 305 or 306. So what are the opportunities for innovation with this research? This model will continue to act as an exploratory tool for researchers. Researchers are able to silence or stimulate certain groups of neurons and then generate data and graphs about how that causes the other neurons in the local circuit to react. And then we can talk about robotics. Robotics and prosthetics for the future will need a human connectome to accurately map neurons in the brain to the prosthetics of the user. Even though we're decades away, a connectome of the Drosophila's ventral nerve cord will act as a precursor to this. As for acknowledgements, I'd like to acknowledge our research group leader, Salil Bade, my mentor, Nico Spiller, as well as the education outreach team, Ilaria and Erica. And I'd like to thank the MPFI supporters and donors, as well as the interns who made this experience very pleasurable. I now open the floor to questions.
Questions for Dylan. If you don't have an headache, just thinking about 5,300,000 rows of...
You don't have any when you look at me. You were talking about how there were so many neurons and they're all identified by numbers, and part of this process was to be able to better identify them. Is there a renaming convention for the neurons once you figure out what it does? Is there a convention that lets you, they don't all name it after the scientists that found it, I hope.
The question was if there's kind of a naming convention of how we rename neurons. It's generally just what the researcher thinks is useful. Like this neuron activates this muscle, so we'll call it front left or something like that.
Was there a specific reason why you chose to make or create a spiking neural network rather than any other type of neural network?
Spiking neural networks specifically model. So the question was why we chose to use the spiking neural network. And essentially, we chose to use the spiking neural network because that has an activation function based on the LIF model. And the LIF model is what most accurately simulates the firing of the neurons without being too computationally expensive.
You could possibly speak another language and I would have understand the same. Any other question? Okay, thank you so much. Great job.